Thanks for sticking around. I don't know anything about sturgeon. This is a really fun project for me because I've been learning so much from what I gather are a pretty extensive group of experts that I'm working with who I think are probably some familiar faces by this point in the conference. It's a really fun project to work on. I should start by acknowledging co-authors on this paper, since I've only really come into the project in the last year. Dwayne Fox from Delaware State University, Amanda and Rich from New York State DEC, Cornell University, Dave Kazak from USGS, and Shuri Sethi from Cornell. So this is a fun project for me because it involves data and Bayesian modeling. But that might put you to sleep, so I'll try to stay out of it. I'm going to start just with a quick generalized life cycle of sturgeon. These not necessarily Atlantic sturgeon, I'm guessing, because it came from the California Department of Fish and Wildlife. But really, the reason I want to start here is what I'm going to talk about today is some modeling work that we've been doing with the long-term data sets that Amanda and crew have been collecting in the Hudson River now for over a decade. So what I'm going to focus on today are the adult life stage when they're up in that spawning area near Hyde Park that Amanda mentioned, and then that juvenile life stage when they're further down in the river in Haverstraw Bay. Give you a little bit of context for this work. These are one of many diatomous species that have experienced large declines coast-wide. Here showing commercial harvest records on the larger graph that's a little bit covered up. You can see a crash down in the early 1900s. That's effectively when this population collapsed. Things went on for another, let's say, 100 years or so, and then by the late 1990s, things started to get even more dire. So if you blow up the far right-hand side of the big graph, that's what's being shown in the little graph here from ASFC. And then after that, commercial fishing was closed, and so those little black lines are going to be dead bycatch records that we have to kind of limp along on there. And so it's just a little bit of context for the work. Some really big reductions here, very heavily fished populations. Of course, there are some other contributing factors to those declines with which many of you might be familiar. This led to a listing for all of the distinct population segments along the coast in 2012, with the Gulf of Maine distinct population segment being listed as threatened and all others as endangered, and this persists through the present day. So, current management status following up on the 2017 ASMFC benchmark stock assessment. Populations around the coast are considered to be depleted. They're also data poor. I meant to search the stock assessment document for the number of times that paucity of data is stated by the review panel and the stock assessment scientists. Some of you actually might know. It's a very common phrase in that stock assessment. When that was conducted, I think there were only 6 data sets coast-wide that had minimal 15-year data sets needed for stock assessments because this is a very long-lived species. One of the data sets that was excluded from that stock assessment because it didn't hit that 15-year number was the juvenile gillnet survey. And there's also an adult gillnet survey that goes along with those. The cool thing is that now, and for the next stock assessment, both of those have reached the minimum term required for use. It's really neat. And in the meantime, people haven't just been sitting around and doing nothing. So this is a graph from a paper that Rich Pendleton and Bobby Adams published in 2021, looking at nominal and standardized catch of juvenile Atlantic sturgeon and gillnets. Again, year on the x-axis here, and the mean annual gillnet catch, if you want to think about it as like CPUE or something like that, there on the y-axis. So these are relatively low numbers. We're talking about, on the order of 2 to 10 fish per gillnet, something like that. Of course, there's variability associated with that. Every once in a while, they'll knock out like a 20 on there, right? And so the goal of this study is to go back and sort of retrospectively see if we can come up with a standardized index for abundance, much like the one that's being shown here. But our hope was to also correct for imperfect detection of individuals. And so if you're into market capture modeling or occupancy modeling, this is a pretty big deal out there in the terrestrial ecology literature. And it's something that we don't always do in fisheries, partly because we survey pretty intensively, right? You don't need to do this if you're doing through-pass depletion, but when you have a big giant river with a few tiny gill nets in it, it's really important to consider those things. And since it hasn't been done yet for the adults, we'd also like to do the same thing for those fish. So in order to do this, we're going to work with long-term gill netting data sets for adult and juvenile Atlantic sturgeon in the Hudson River. There are little tiny boxes here that you might not be able to see from where you are. One is down in Haverstraw Bay, and you can kind of see a pile of dots there. That's this pile of dots. There's a lot of dots. There's like 3,000 dots in that map that we're going to use today. And the one on the left is the Hyde Park reach of the river. That one's got like 700 dots in it. So these are really cool data sets. I should note that there's been a lot more extensive survey work done in the upper river for adults, but we wanted to trim the data set back to what was represented across the time series. The adult survey is conducted from did I reverse those? I don't know, February to May, let's just go for it. Dwayne said yes. From May to June, and the juvenile survey, students don't start your talk at 7 a.m. the day you have to talk. The juvenile survey is conducted from usually March to May, starts a little earlier in February some years. We're going to split these reaches up by shoreline and then by river kilometer, so we'll end up with about 10 combinations of sites and river shore for the juvenile survey, and 12 for the adults. We'll go west shore, middle, and east shore there. And what we get out of this are really cool pictures that Dan has to ask Amanda for. This is a picture of one of the gill netting surveys, just giving you an idea of what's going on. My understanding can be pretty touch and go depending on conditions, right? So when you're thinking about this from an occupancy or market capture framework, you've got to try to account for those kinds of things. So what we're going to work with, we wanted to work with initially was counts of fish in these gill nets. And we try to implement those with an N mixture modeling framework, but they're so variable that might have exploded in my face. And so now what we're going to boil this down to is slightly lower resolution information. We'll be working with ones and zeros. Did we catch fish in these nets or not? But we're not going to completely disregard the counts because we got them. And if we can estimate detection, we could estimate population abundances within these local surveys. How do we do that? Aren't you lucky? I'm not going to go through this entire prep. This is for those of you who like this kind of stuff. But basically what we're going to assume is that Y, the catch or failure to catch sturgeon, is the outcome of coin toss with some probability of detecting fish at all. We're then going to, based on that probability of detection, assume that we can derive a probability of detecting individual fish within those nets, given that they are present. We don't know how many fish are there. That's big N. So what we're going to do is to start this whole thing off, we'll take a guess at that. And then we'll say that that's a function of survival S and the number of gains to the population G. Each of those is going to be given a loosely informative prior just to keep things in the world of estimable parameters. And then we'll do a couple of those things on the log scale. So effectively now what we've done is we've taken a repeated count survey, boiled it down to ones and zeros, and then we're going to force a model to estimate latent biological parameters that we can use to inform local abundance estimates while accounting for imperfect detection. That was way less painful than it could have been. Cool, results. All right, I'm going to present 3 types of graphs. This is the first of those types. We probably don't need all this information, but I'm a modeler, so I want you to see what's going on, warts and all. So in the top left here, you have this little letter omega, is going to be our joint probability of survival and remaining in the study area the following year. This is for juveniles. Relatively high, as one would expect for a long-lived species. The fact that its median is down around 0.8 just means we have some emigration, we have some recruitment to adulthood. We haven't been able to quantify that yet, but we haven't given up. The next one down on the bottom are gains to the population. So anywhere from a fish or so a year to a little bit higher than that. So relatively slow immigration or recruitment to the juvenile survey is what you're looking at. The really fun rainbow color graph in the top right is showing the probability of detecting any fish in a net. And all of those different colors are our different sites. So you can see we've kind of got a shotgun blast there. And you can see two things that might become apparent is, one, maybe I should have gone with a grayscale. Well, two is it kind of bounces all over. If you really squint, you can see that that detection probability has come up in more recent years. My guess and my strong suspicion is this has to do with getting better at fishing for sturgeon and knowing where they are over the years. So it's kind of important that we account for those kinds of changes over time if we want to get standard indices of abundances. The lower right graph with all of the colors is that individual detection probability given that the fish are present. And you can see we're pretty low, down around 10% chance of detecting a fish, an individual, when they're there and vulnerable to the gill nets. These are the kinds of biological parameter estimates we get out of them. I should have mentioned before that the lines are our median estimates, the ribbons are 50% credible intervals, and then the vertical lines are 95% credible intervals. You can call them confidence intervals if you like. So we have relatively similar local abundances to the juvenile, nominal, and standardized catch indices. That's good. That means we're doing a decent job detecting fish when they're there. You see that there was a slight increase from the start of the survey up until about 2014, and then after 2017 or so, that starts to decrease, and that's congruent with the standardized and nominal catch indices that I presented in the introduction. On the right, we have the number of surviving and remaining in the study area. That follows the same pattern, of course it does, right? And then on the bottom, you can see that I've got some really squished graphs that don't look like they mean a whole lot. And we're a little bit perplexed by that right now. It might just be that we have low immigration and emigration rates. It might be that the model's doing a poor job estimating those. From all of that, we can take our site-specific or local abundances, correct those for individual detection probabilities, and estimate a reach-wide abundance for Havistraw Bay. You see we're bouncing around between about, 150 and 450 fish in a given year, and those trends line up reasonably well with the nominal catch index. The points are our estimates, the vertical lines are 95% credible intervals, and the dashed line is the nominal catch index that Rich and Bobby put together. So the patterns more or less follow the same thing. I should note that those are on very different scales. So we aren't estimating fewer fish than the nominal catch index. It's just an artifact of having two different data sets on one graph. Okay, now hopping into the adults. Really similar stuff here going on in terms of the survival and retention probability in the top left. So again, median somewhere around 75–80% probability of surviving and staying in the study area to the next year. On the bottom, that's numbers of gains and losses. Again, really similar to the juvenile index there. And in the top right, you see a very rapid increase in the probability of detecting fish until that levels off. And that one almost certainly is related to having found the fish, using side-stand sonar to kind of focus the surveys over the years. But it does level out pretty nice. Some of those bounce around, right? There's a lot of site-specific variability here. But overall, this sort of nice increase to about a 50-50 chance of detecting a sturgeon if they're there at all. Same thing as juveniles in the bottom right, fairly low probability of detecting any given individual. And if we look at our derived quantities, we see that we've got a slight increase from the start of the survey through the end of the survey, most recent year 2022, and that seems pretty consistent across sites and years. That's a bright light on the horizon, maybe, we think. Same deal going on with the other parameters. The big takeaway from the bottom two is that I don't think we're doing a great job estimating biological parameters, or they're on such a small scale that it's really hard to distinguish it from noise at this point. Really what we care about from this is that individual detection probability, because now we can estimate a reach-wide abundance for spawning Atlantic sturgeon in the Hyde Park region. Now, I mentioned before that this is an estimate of those fish that are vulnerable to the gillnet survey. It's really cool because this is downstream or including the major spawning area, so we think most of the fish that spawn in the Hudson River pass through this survey during the time that it's conducted. So we think this is a decent approximation right now to the spawning abundance. And the one thing that I'll point out that's a little bit different here is we have a gray dot, and that's work that Dave Kazak did as a postdoc to estimate a abundance of spawners in the same area of the river, totally different methods, and they're within like 15 of each other, which could be totally coincidence, but gets people like me really excited. You'll notice our uncertainty is a lot wider. I'm okay with that because it doesn't go up to like 100,000 like it did before. So big takeaway, we've got some detection corrected indices for juveniles and adults. We've got increases in detection probability over time, which suggests that we're surveying more efficiently. We see some increases in the juvenile abundance through the mid-2010s, with some decreases thereafter, and it seems like the adult abundance continues to increase. I'm not 100% comfortable with the estimation of the biological parameters, immigration, but we're still working on those. Does this work? Well, I'm not sure yet. We're going to do some validation. I once heard Todd Arnold, ecological modeler, say, you can do that, but you shouldn't do that. So I want to make sure that we can do this. So we're going to be doing some simulation work this year, to validate the approach. All of the nuts and bolts are validated. I just want to make sure everything's tied up with a nice bow there, really. And then we'll be doing some exploration of sampling design requirements to determine how much sampling and for how long we would need to do it to really start to rein in those estimates. With that, I'm fully out of time, and I would like to acknowledge all of our home institutions and Cornell and DEC for funding this project.